Dr. Gable is an associate professor of neuroscience at Washington University in St. Louis, and he's a leading researcher in the field of neurodevelopmental disorders. His lab focuses on understanding how genes like DNMT3A affect brain development and function. As you all know, DNMT3A is the gene responsible for Tatten-Brown-Raman syndrome. Dr. Gable has been a dedicated collaborator in our efforts to better understand Tatten-Brown-Raman syndrome right from the start. He is an esteemed member of our scientific advisory committee, from supporting our summit to helping design studies that bring us closer to treatments. He's deeply committed to making a difference for families like ours. We're really thrilled to have him here to share updates on his work and some more information on gene therapy and TBRS and the future of TBRS research. So I'm honored to hand this over to Dr. Gable.
Great. Well, thank you, Jill. Can you hear me okay? Great. Is that sharing appropriately, or are you seeing, yeah, all good? Okay. Yeah, well, thanks. When you contacted me, I too saw the recent article in the New York Times that I'll touch on briefly, and has made waves in the gene targeted therapy world. And so it's exciting. And it made perfect sense to try to explain what it means, and then we've been thinking for a while now about gene therapy as it might apply to Tatten-Brown-Raman syndrome. So I'm happy to describe where we are. I would say that this is, although I have a lot of slides, because scientists makes lots of slides and I couldn't stop myself. This is intended to be informal, and please, interject if you have questions or something is unclear. Hopefully I will not spend more than a half an hour or so talking about the details max. So there's time to interject if folks can stick around, and I can stick around at least for an hour here. So really just trying to be here to answer questions above everything else, but hopefully some of what I've got here will answer questions that you have or didn't know you had, and now we'll have information. So any questions before we start? Otherwise, I'll launch into it. Okay, so I'm Harrison Gable. We're here today to talk. I'm at Washington University in St. Louis, and we're here today to talk about gene therapy considerations in TBRS. And I think it's considerations because there's still work to do to know if gene therapy is something that's appropriate for TBRS. It's certainly something that is applying more and more to rare diseases caused by genetic alterations, but there are things we need to learn. So, but it's an exciting time for these types of therapies, and TBRS is on the radar screen, and I'll tell you about our work and work from others that's continuing to push that. So first off, who am I? I'm Harrison Gable. As Jill introduced me, I've worked with the TBRS community for a number of years now. I'm a preclinical researcher, and I really started off studying basic molecular mechanisms in the cell that I'll try not to touch on too much today, because it would be, although I find it very interesting, not necessary. And then we study neurodevelopmental disorders, particularly disorders that affect the brain, the development of the brain, the function of the brain, because it turns out that this epigenetics that I mentioned we work on is a jargon term to describe actually something that's disrupted to often cause neurodevelopmental disorders. I've been working on DNMT3A, the gene, for about 10 years now in my lab to understand what it does in the brain and in brain cells. And that led us to understanding how TBRS occurs, and particularly its impact on the function of brain cells and neurocognitive issues in the disorder. But more and more, we're working across all the tissues that are affected across the disorder. We collaborate with a ton of different labs to do studies really in the preclinical space. That means understanding genes and the effect of disruption of the DNMT3A gene. But we're more and more working with clinicians and trying to basically bring as many people into a really big TBRS research tent to cover all aspects of the disorder. And then I'm an advisor to the TBRS community, really as a preclinical researcher. But I bring to that a lot of experience I have in Rett syndrome research. And as I'll mention briefly, Rett syndrome has been sort of a bellwether for gene therapy. And so I have that insight to bring along and to help us think about things like gene therapy and TBRS. One thing I have to say very clearly is that I'm not a physician. I'm a PhD, a fake doctor, as my niece will call me. But I am preclinical researcher and really an expert in the underlying genes and the aspects of the disorder that are affected in the disorder. And at this point, gene therapy is in that space. Gene therapy for TBRS is in that space. It's really about understanding the fundamentals of the basic biology of the disorder and can we treat it. Unfortunately, that means that I cannot provide treatment advice right now. I'd be happy to refer you to great physicians, including some physicians that are on the call right now, to think about what you might need there. But this is information about where the research is and where we might be heading. And I just want to be clear about that because I don't want to be suggesting that I can give you a treatment advice. Okay, so we'll launch in, and I'm going to start by giving you sort of an idea of what gene therapy is and how it was recently applied in a very exciting and sort of triumphant way to an individual patient, a new type of gene therapy. But what is gene therapy to start? Well, DNMT3A is a gene, and we'll talk about that in a second. It works to help your cells function, and when it's disrupted, a mutation of that gene causes Tatten-Brown-Raman syndrome, which we're all very familiar with. Gene therapy is about replacing the function of that gene. So this is the definition, and there's actually this website that's listed here is really a great resource for understanding genetic terms and molecular biology terms when you hear about them. And it defines in a very simple way that gene therapy is a way of using genes, DNA or gene products, to prevent or cure diseases. And usually it works by fixing a gene or replacing that gene. And that's what we're talking about potentially doing in TBRS if we can get it to the point where it's effective and safe enough. Because if a disorder is caused by a broken gene, then fixing the gene might be a way, in many cases, to really make it a lot better, rather than trying to treat the downstream consequences of the loss of that gene. That's not always the case, and we'll talk about that a little bit, but often it may be the case, and TBRS is certainly a candidate for that. So why is there so much excitement about gene therapy? Well, there's a very high profile. There's been quite a few successes in gene therapy recently in rare disease, but there was a very high profile article that came out in the New York Times and made it across the pond to Europe and other places, talking about a really great success story. KJ is this little baby, as we see here, and this is the text that my friend sent me, excited about gene therapy and knowing I work in this space. So KJ has basically a baby with a severe disorder that's been treated with what's called a gene editing technology, a type of gene therapy, and been made a lot better in this sort of Herculean effort that demonstrated the power of gene editing therapies. So I was going to go through briefly what actually happened with KJ and to introduce you to the idea of how we would do this type of work, and maybe to answer questions that might have come up if you saw the article. And then we will sort of transition to TBRS and what it might look like for TBRS in the long run. So KJ had CPS1 deficiency, like TBRS. It's a specific gene that's disrupted to cause this disorder. It's a disorder in which the liver doesn't function right and can't process, do metabolism right, and ammonia builds up in the blood because ammonia cannot be converted to urea, which is then cleared from the bloodstream. So you end up with way too much ammonia in the blood, and this can have really severe impacts on multiple organ systems. It can be fatal, and it can cause, even if it's treated, can cause neurological damage that's incorrectable over the course of the lifetime of the individual. So an extraordinarily severe disorder, and it's a very rare disorder caused by mutation of this gene, sort of breaking both copies of the gene. So very rare and very, very severe, two aspects that made it a good target for this type of new approach. But it needed, in order for KJ to be treated and really to be healthy, he needed to be treated very fast because basically the more months that go by, the much more severe KJ is going to be. So a consortium of scientists at University of Pennsylvania and CHOP, together with really a group of pharmaceutical companies, put together this Herculean effort to, in a few months, develop a bespoke gene-correcting therapy that could fix the gene in KJ's liver and help him have a functional liver again. And this is a very simple version of what they did, and it's leaving out tons of work, but they basically created a little package that can go in and fix the DNA. It's called CRISPR technology. You may have heard of it. It won the Nobel Prize. They created a bespoke version of that, and they put it in these little particles called lipid nanoparticles, which, when they're injected into the bloodstream, will go into the liver. That's one of the things that made this very possible. And then they put that little cargo in, they injected into KJ, it went to his liver, and it corrected the gene enough that there were enough cells that could do the job and fix his disorder, basically. So, this story is really remarkable because he, for example, went from about the 77th percent in body weight, I believe, back to about the 50th percentile, right? So really, and you saw that picture, he's a nice, fat, healthy baby boy. So really a remarkable triumph. So why is this such a triumph? Well, one, obviously we had one child who had to some extent what might be considered a death sentence, perhaps not cured, but brought back to an incredibly wonderful level of health. And that's a triumph in and of itself. But it was also the speed with which it was developed, which demonstrates that we have all the tools we need to do this, the efficacy and the safety of the treatment so far, which is something that needs to be watched. And all of that together has really been held up as a sort of a demonstration that all this basic research and all this building towards being able to do things like this gene therapy is actually now coming out and possible. And it sets up what we would consider a platform that can be used over and over again in this context. Now, it needs to be made more efficient and more feasible. This was a Herculean effort, and it's not really possible for one individual patient on a daily basis to do this. Because it's too expensive and complicated right now. But once you do it once, you can get better and better and better at it, and you can work towards doing it for as many patients as possible. Which brings us to, does this change the game for the TBRS? And I would say the answer to that is both yes and no. Yes, for all the reasons I just described. Basically, once you demonstrate in science and medicine that you can do something and you can do it efficiently, it powers more of this work, and it gives us a map for what we can do. And that's being held up at the end of this scientific article that described the treatment of KJ. They said, we expect that this we'll be able to do this for many additional rare disorders because they've shown it's possible. So is that a game changer? I think that is, in that it gives us a new weapon in doing gene therapy. There are other forms of gene therapy that are already out there and effective, but this is a new weapon, and it's really, really, really exciting. The no part of it is unfortunate in the sense that although you can make great advancements in one disorder, you need to understand your disorder, and there need to be the right combination of understanding, opportunity, and really the biology needs to give us a chance to be able to do gene therapy and be effective. And all of those things still need to be worked out for TBRS before we can go ahead with the gene therapy. So that's the frustrating no part, but it's a reality that you should know, that it wouldn't be possible to just go ahead right now. Even if we could do it, we're not sure we need to do it, and it's worth the risk. And so that's where we as preclinical researchers and then clinical researchers are working really hard to try to solve this. So that's an overview of broadly sort of what this news story tells us. I was now going to jump into what very briefly what DNMT3A is and what it might look like to do gene therapy for DNMT3A. Okay, so very briefly, I want to introduce you to the idea of what is DNMT3A. It's the gene, as we've described, that is disrupted or mutated to cause Tatten-Brown-Raman syndrome. What does DNMT3A do? Well, all the genes in your body, they really sort of are the blueprints for little molecular machines called proteins that work inside your cells and have jobs. And we won't get deeply into what DNMT3A does. I can talk about that ad nauseum. That's a lot of what I work on, but it's not so important here, other than to say that in all cells in your body, in particular in brain cells, it has an important role in basically modifying the DNA inside the cells. It puts something called DNA methylation on the DNA inside of the cells, inside the nucleus of neurons, for example. So you're inside the brain, you're inside a neuron, a brain cell here, and then inside there's this little molecular machine known as DNMT3A protein that is putting this other chemical mark on the DNA. and it's helping the cells basically decide to make the right choices on what they need to do on a daily basis. And so, and that role is important in many, many cell types. My lab in particular studies in the brain because it does something particularly special in the brain. We think this is in part why you get neurocognitive challenges when you have TBRS. So that's what DNMT3A does. So that's the protein for DNMT3A, and it's produced. I have an animation here I forgot about. I apologize. So it's produced. Let's see. Hold on. I'm going to very quickly de-animate my slides. See if this works. So that protein is created from a gene, from the blueprint, the DNMT3A gene. And so this is the molecular biology by which that occurs. The DNMT3A gene, which is a sequence of letters inside your DNA, makes the DNMT3A protein through this process, which we don't have to get into the details of. And under normal circumstances, you have a DNMT3A gene that helps produce this protein. The variants or the changes that we were just talking about cause a mutation in the gene that I failed to animate in the right way. They cause a mutation in the gene, a change in the sequence of the DNA that then leads to a broken protein. I apologize for doing this on the fly. I realized I couldn't animate if I was going to share my screen the way I was going to do. Okay, so instead of having a functional gene, there's a break in the sequence, and that breaks the function of the protein. So now we don't have enough of DNMT3A in the cell, and that's what drives TBRS. So this is what it looks like in TBRS. You have a gene. So in TBRS, the DNMT3A gene is broken. The last part you need to know is that there are 2 copies of every gene in your body, one from your mother and one from your father, and only one of those copies is broken in TBRS. So in a normal person, they have 2 DNMT3A genes. Those DNMT3A genes produce enough DNMT3A protein to go do this little molecular machine job in the cell. And in TBRS, one of those copies of those genes is broken by a change in the DNA sequence. And that leads, we think, by and large, to just a loss of function, we call it, a loss of about half of the functional protein to cause TBRS. So the model for gene therapy would be to replace that missing gene function, right? A gene therapy would go in to somehow deliver one approach would be to deliver a new copy of the gene totally separate from your own DNA. And this is sort of conventional gene therapy where you actually load a gene into a virus, for example, you deliver that into the body, and it adds back the activity that is the functional DNMT3A protein, even though there's still a broken copy of the gene in the cell. And this is sort of classic viral gene therapy where you deliver a piece of DNA that has the gene that can then get inside the cells and make protein. Can also be, we'll talk about the different ways to deliver it. KJ's therapy was slightly different, and where basically little molecular machines, CRISPR, is delivered into the cells, and actually it's targeted to go fix the DNA of the individual themselves inside their cells. So instead of replacing the gene, you're fixing the gene. And this is a little bit more work if you have to fix different breakages throughout the gene, as was just discussed. But in some ways, it's better because it will put back exactly the right amount of the gene. Whereas if you add back the gene with a virus like this, you could potentially add too much or not enough. And so that's the upside to this approach, is that you're fixing the gene in place, and so it knows exactly how much to do its job. So that's broadly the concept behind how we would potentially do gene therapy in DNMT3A disorders. I'll talk about a couple of different approaches in a second here, but I wanted to lay out a roadmap for what would need to be addressed to think about gene therapy for TBRS. And I'll talk a little bit about where we are. So here's the hurdles, and this was one question brought this up, timing. When do we need to fix the gene in order for a therapy to be effective? And for a developmental disorder, a number of years ago, we thought that it wouldn't matter if you put the gene back because development had already happened. And it might not be possible to correct the missing function of the gene that was needed in development. I'll talk a little bit more about that, but in a lot of disorders like TBRS, we actually realize that the ongoing function of the gene might be really, really important, and you might be able to correct an awful lot. Then we need to get the right amount of the gene back in. I just talked about this briefly. I'll talk a little bit more about that. But we need to know how exact you need to be in terms of delivering the gene in order for it to be effective. Once we know all that, we have to come up with using one of the tools that's being developed to bring that to TBRS to do it in just the right way. Do you correct at the right time? Do you have to have the right dose, or do you want more or less of a dose? And that's the approach. I'll talk a little bit about that. Then how do you deliver it? Do you have to get it to all the cells in the body? That's a tall order, right? To get it to every single cell in the body. And TBRS does affect most, if not all, organ systems to some extent. But we may have to make choices about where we can deliver it really effectively. And we want to deliver it to the places that need it the most. We talk about delivering it to the brain because of the neurocognitive challenges. There's things like cancer risk or overgrowth, other things that we might lead us to go for one tissue over another. And then finally, we have to make it safe, which is, a big challenge and really a limiting factor in applying these gene therapies. And I'll talk briefly about that. Okay, so the first question would be timing, and does adding the gene back in a developmental disorder ever help if someone's already born and they've already gone through development? Well, there's a really prominent example that has shown that in even really severe neurological disorders that were considered neurodevelopmental, that you can add back the gene and will have a big effect. And this has powered the idea that we could do gene therapy for disorders like TBRS. And this story starts about 20 years ago. Researchers were studying Rett syndrome, which is a severe neurodevelopmental disorder, which is molecularly related to TBRS, but looks quite different. A disorder in which largely girls have the disorder because the gene is on the X chromosome. But those details aren't particularly important, but it's very severe with individuals with the disorder sort of losing controlled hand movement, speech, having severe seizures, often wheelchair bound, and it's caused by mutation of the MECP2 gene. And researchers were studying this gene and what happens in Rett syndrome. And although there were really severe symptoms in the disorder, they noticed that the brain was intact in the individuals who had the disorder. So all those cells were still there, even though they had really, really severe effects. And they made a mouse, like we tend to do these days, to study what goes wrong in a whole organism when you break the gene. So they broke the MECP2 gene. They got a mouse that had the mousy version of Rett syndrome, this is what it looks like. Here are two normal mice. And then if you're not a mouse researcher, you might not find this too striking, but this mouse is very, very sick. It has trouble breathing. It has trouble walking. It has all the aspects of the disorder. It's what we would consider a very good mouse model in that it replicates many aspects of the disorder. In this case, because for some details, the mouse is so sick, it's actually going to die at about 8 weeks of age. So we've created a mouse that has Rett syndrome effectively. And this is largely work done by Adrian Bird, a researcher in the Rett syndrome space who actually spoke for the TBRS community CRN a couple of years ago. So we have this very severe mouse, and the question is, and it's very severe disorder, and the question is, if we put the gene back, will anything get better? Now, Dr. Bird looked at this mouse and said, it's got an intact brain. All the cells are there. Maybe it just needs the gene back and it will get a lot better. And the individuals with the disorder, we saw the same thing. So what he did is he made a mouse where he could start with the MECP2 gene broken. It has Rett syndrome. And then he could use a genetic trick together with some pharmacology to turn on the gene. This is not delivering it through conventional gene therapy. This is literally just being able to switch the gene back on and say, if I were to make perfect gene therapy, what would the effect be? So this is a mouse that has this gene that he can fix before he fixes the gene. And you saw those mice before running around the cage, sniffing. They'll run away if you give them a chance. This mouse is breathing heavily and actually in an irregular way if you look closely, and it's very sick. It has Rett syndrome. Then he turned the gene back on. This is in an adult mouse, to get back to the question of timing. Oh, I should say this mouse also has very severe neurological symptoms where it cups its hind limbs together in a way that suggests it can't control its limbs very well. He then turned the gene back on in the same mouse and asked, does the individual get better? Does the mouse get better? This is the same mouse a few weeks later. Now, if you're a mouse biologist, you say this is a very healthy mouse. In fact, I work with mice, and I wouldn't necessarily be able to tell you that it's different than a normal mouse. And that was a mouse that would have died if he had not turned the gene back on. It, the mouse, by many, many measures, almost all measures we can come up with, comes back to very, very close to a normal mouse. So this is an incredibly striking finding because it says, at least in this disorder, if you put the gene back, you can correct a lot. And for example, this mouse now does the usual thing with its hind limbs. It doesn't actually sort of do this clasping motion, but actually has much better neuromuscular function. So this was an extraordinarily striking finding. This happened about 20 years ago now, and directed gene therapy towards neurodevelopmental disorders, even disorders where the individuals can't speak, they're in a wheelchair. This is the mouse equivalent of dramatic improvement, maybe walking after being in a wheelchair. Now, it's in a mouse, but it is a proof of principle that you can potentially correct a lot if you put the gene back. Now, there are aspects of Rett syndrome that suggest that it might, and that I won't get into the details of molecularly and in cell biology, that suggest it's particularly fixable in this way. So I don't want to say that every disorder, including TBRS, is as fixable as this, but it changed the way we think about developmental disorders and told us we needed to do the experiment. So the thought is we need to go do this experiment in TBRS and see whether or not we can have a positive impact when we turn on the gene. So now I'll start telling you about aspects of gene therapy that we're at least starting to work on, that is exploring this type of work and doing this type of work to look into, is gene therapy feasible for TBRS? So one set of studies that we're doing in my lab is basically this exact same set of experiments. For those of you that follow the mouse research and literature, of course, TBRS is not as severe as Rett syndrome. But of course, it has many severe symptoms, and we have a mouse, we have several mice actually, that we can study that have the mouse version of TBRS. And we've gone and we've, using another mouse actually, engineered the same setup where we can turn the DNMT3A gene back on in a mouse that already has TBRS. And to the point, the question about timing, when do you need to do this or when can you do it? We're asking that question by turning the gene on at different times and back on. So we're basically mimicking sort of perfect gene therapy. The gene comes on perfectly in all the cells in the body. And then we're asking, how much better does the mouse get? What I can tell you is that in early returns, when we turn on the gene right after birth, we see that we can correct some of the molecular deficits that we know occur in TBRS. So that window suggests that some things will be corrected if we could put the gene back. That is very, very early, sort of unpublished data. But there's some biology that says that we think we're going to be able to correct if we can get the gene back on. So that's hard work that we're currently working on. There's additional investigators, many of which I probably don't know about because they're in different parts of the world. They have funding from different places, all working on this idea in TBRS. Another example of this type of work is work done by Timothy Lay here at WashU. He studies the disruption of DNMT3A in cancer. As many of you know, that certain types of disruptions are associated with cancers that occur later in life and not necessarily with TBRS in the first place, but there is some cancer risk, we believe, in TBRS. And so he's been asking, can you put the DNMT3A gene back in blood to avoid the cancer risk, or in the case of people who have cancer, to avoid cancer? And the way he did this was to deliver the DNMT3A gene into the blood cells, and transplant in fixed progenitor cells in the blood. What he found is that when you did that, you could actually really correct a lot of the molecular deficits in the blood. Another example, so that would be very late, even in adults, where you can, in certain tissues, you can correct issues that are there. So this is encouraging, and that suggests that in some tissues, correction even very, very late is going to be helpful. Of course, we have to sort of balance the efficacy and the danger of doing these types of therapies as they're being developed with how much they help, but this is encouraging. And he did this with a viral approach where he put it into the blood cells using a virus, and basically got the gene to turn back on, and this DNA methylation that DNMT3A does to get back on. So a little bit of evidence suggests that the timing is going to be important, but that there may be stages throughout life at which adding the DNMT3A gene back would be helpful. The next big challenge is the dose part, right? So TBRS is caused by missing one copy of the DNMT3A gene and having half as much of the protein around. Suggests it's already dose sensitive because you have some working DNMT3A even when you have TBRS. And this is quite common in a lot of neurodevelopmental disorders that are caused by just missing half of the gene. Turns out that if missing half of the gene is detrimental, that sometimes having too much of the gene is also detrimental. So, there are disorders there'll be disorders caused by loss of a gene, and then there'll be duplication, or too much of the gene can also occur and cause a different disorder. And if that's the case, then you don't want to deliver too much gene through the gene therapy because you could actually drive a different disorder if you do that. And for TBRS, there's some evidence that this is something we should really care about. There's this disorder that we think is caused by too much active DNMT3A, although the mutations that cause it aren't exactly just too much DNMT3A. But there's also some evidence from mice, if we have too much DNMT3A activity, that there could be detrimental effects. This is unfortunately not a good thing for developing gene therapy, because it would be ideal if you could just add as much DNMT3A as you could, and it would just make you better without having to worry about overshooting. But this is sort of what it would look like if we add with viruses. This is a little virus particle. We add too much DNMT3A, it might start doing things it's not supposed to do, or too much of a good thing, it will become a bad thing. Now, there are ways to try to think about avoiding this, and correction of the genes that are in the DNA rather than adding more DNA is one way to do that. Also, work by Tim Lay in that same paper that I showed in the previous slide is working to see if there's ways to turn up the DNMT3A that's still around, because in TBRS there's one good copy, and turn it up so that you can't overshoot. There was a method he used that sort of had another little molecular machine in the cell help turn up the activity of the DNMT3A protein that was there. So we're exploring those types of options because we think dose sensitivity might be something that's important in TBRS. Okay, so those are the considerations we're working on, and we're really in the stage of trying to understand what the best approach would be. Once we have that information and we're watching other disorders do this work, you then have to decide on what's the best approach. And this is just a small menu of the types of approach that you would do. I've sort of covered them very briefly, but you could either replace the gene, deliver more gene, like just more of the DNMT3A gene, the entire thing, with a virus or these lipid nanoparticles, and then it can correct. But as we talked about, if you do that and you overshoot, that could be bad. So you have to think about getting just the right dose of DNMT3A into the genome or into the cells. The other way, which was what they did with KJ, is to fix the mutation in the DNA. This is good because you can't overshoot. If you fix the mutation, then you just have what your cells are supposed to have in terms of the total number of DNMT3A genes, and you can't overshoot. There are challenges here, though, because you have to maybe make a different slightly different type of therapy for each individual that has a different mutation. And this is a lot of work and requires sort of safety and efficacy development for each individual. Now, KJ shows that we can do it, but we have to make it fast and reliable and safe, and sort of scalable. And that's the next big challenge. So each of these approaches have different pros and cons, and basically the field is developing these, and we're looking at what's being done and going to be trying them out in different ways in different labs, starting probably in our mice. I just want to briefly mention there are also other ways to do this, and I apologize, this slide is pretty technical, but you don't necessarily have to replace the DNA or put new DNA in. Sometimes you can take an intermediate molecule called RNA. So when you go from the DNMT3A gene making the molecular machine DNMT3A protein, it goes through something called ribonucleic acid, or RNA, and that is being produced, and the amount of that and how much protein is produced from that RNA can be something that you can change. So there's something called antisense oligonucleotides that can go and change how much the RNA is used. This is the mechanism by which one of those approaches might work, but basically you put in a little piece of DNA into cells, and it goes and helps the DNMT3A that's useful, that's still there, work even better. basically make more of the DNMT3A protein, even though you don't correct the gene, the mutation that causes the disease. So these would be called, these are RNA targeted therapeutics using antisense oligonucleotides. There are other approaches to try to target the RNA that are being explored for a lot for Rett syndrome. And when what they learn in Rett syndrome, we can bring to our field. And I'm actually collaborating with some WashU researchers who are trying to come up with this as a approach for many neurodevelopmental disorders. And we're going to be helping them look at TBRS once they develop these types of approaches. So there's a sample of some of the approaches. I could talk to you all day about all the different approaches that are being explored. And the great thing is, companies are developing these. So the hope would be that the ones that work best will be ready to use if and when we can show that a gene therapy for TBRS works well. The last challenge that I'll just touch on briefly is getting the therapy into the right cells and to enough of the cells. And this is, again, an area of development that sort of doesn't apply specifically to TBRS, which is great because there's companies and researchers developing these approaches. With KJ, it was something called lipid nanoparticles, basically a little blob of sort of cell membrane or something that looks like a cell membrane or a virus, but it's not actually a virus. It's bioengineered that you inject into the body somewhere and it will go into the cells. That works really well right now for the liver, but it doesn't actually get into the brain, for example, in a way that would be efficient enough that we could use it as a treatment if we wanted to try to fix the brain cells. Viruses, on the other hand, can get into the brain and get into neurons and get throughout the body. And so that's another approach. Basically, we hijack viruses and we use them for good and deliver therapies that way. Once you develop these in concert, you're going to decide where you're going to deliver it. And of course, this comes down to the symptoms and the challenges in TBRS we want to sort of most directly address. Of course, neurocognitive challenges are a big thing. So, in the long run, do we want to be delivering this to the brain? If we think it can have a positive effect, we may in the long run really want to be trying to do that. And different companies and groups are developing ways to try to deliver to the right cells at the right time. But that's ongoing. And for things like treating the brain, that's a pretty big challenge. Rett syndrome, again, is a place where we're getting a lot of information, and there's gene therapy trials going on because of that study that I showed you in Rett syndrome. And we'll be getting initial reports on how well it's working, again, at a conference I'm going to in a few weeks. From a safety and delivery standpoint, we'll learn more about that. And whatever we learn in Rett, we'll lay the groundwork for potentially doing this in TBRS if it becomes something that we think will be really helpful. The last part, which is, of course, cannot be ignored and should be paramount, is safety. And one of the big challenges with gene therapy is you can make these, we could inject a gene, we could create something to deliver to the gene to many cells in the body today, or at least with, we can do that. But if we deliver that and it causes severe, severe reactions or on its own causes such a detrimental effect, it's worse than the disease, then of course we can't do that. And with gene therapy, there's real risks here. So the treatment can't be worse than the disorder. For example, delivery of the gene with a virus brings along with it a risk of a very severe immune response that can at times be fatal. And actually in the gene therapy trials for Rett syndrome, one patient who was getting virus was actually sadly died. These are immune responses that we're learning how to control. But that's a risk that you have to weigh against the potential benefit. When we're doing this genome correction, there's a small chance that you could go change other parts of the genome, and that can cause cancer. So we have to consider that when we're engineering the genome, that there can be detrimental effects. Delivering too much of the gene could be detrimental, as we talked about. Other challenges are, particularly with viral gene therapies, if you do deliver it, you can only deliver it once, because it would cause a severe immune response if you did it again. And so you basically you get your one chance, and then if it doesn't work or if it overshoots and has a detrimental effect, you may be sort of stuck with that. And that's therefore really important to try to make sure we avoid that kind of a challenge. So this all comes down to, for a therapy to go forward, it has to be safe enough, and all those risks have to be mitigated enough, and the upside has to be good enough that when weighed against the normal course of the disorder, the upside and the low risk has to outweigh the normal course of the disorder. And in TBRS, although it's we all know how challenging it is and want there to be therapies for it. In terms of where we are with gene therapy, it doesn't quite get to the level of severity and the level of what we think correctability that companies are willing to dive into it yet. Instead, they're going for disorders where there's either, where it's so severe that if you don't do anything, it could lead to death. For example, with KJ. So they're working in spaces where there's either such danger of severe outcomes that you can move very fast and you can try things, or you can keep it safe and you can keep it limited. For example, gene therapies in the eye, where you can control where the virus goes, and you can basically sort of keep the risk to the rest of the body limited. So right now, those are the places sort of where you can either limit the risk, or the risk is so high without treatment that it's worth doing this. And TBRS, unfortunately, right now is not in that space. So it's sort of a challenge for researchers to get to the point where the risks go down, the delivery gets good enough, and our potential upside gets high enough that compared to the rest of the disorder, we're ready to go and we can justify it. So where are we then? Well, the challenge is that we have to get through all of these hurdles, and the blessing of TBRS is it's not fatal on its own, and therefore it doesn't quite get to the front of the line on these really risky gene therapy trials. The downside, of course, is that then we're going to have to go farther before we can go ahead and be trying gene therapy. And we got to get through all these hurdles before we're ready to do it. And KJ's story and these new technologies are certainly pushing things faster and telling us it might be possible, but we're still marching through those things. So that's the challenge. The good news is we have many labs, and more every day. I try to get as many folks here at WashU working on TBRS as possible, and I know there's lots of other folks doing the same. Many labs are working on the disorder and trying to tackle each of these issues. And we're not alone. Gene therapy for rare disease, like in KJ's case, are having advances, and an advance for one disorder where you fix a gene is actually advanced for all disorders, because what we learned there can be applied to the different genes. And that's why KJ's story is so exciting. So if we look at these hurdles that we have to get over and before we can do gene therapy, we can sort of think of the front end of this is where TBRS specific researchers need to be doing the work. Does putting the gene back after birth help? My lab's working on that. Do you need exactly the right amount of DNMT3A for it to be better, for improvement? And do you need to worry about overshooting? Labs are working and thinking about that. And then as we try to do that, we're sort of taking the latest approaches and trying them in our mouse models to show, hey, look, it could work. And this altogether is, the phrase is de-risking the possibility of gene therapy for TBRS, so that biotech and pharmaceutical companies say, oh yeah, this is worth trying. And once we do that enough, then we can we can really lean on this remarkable progress that's being made with nationally, federally funded research and biopharmaceutical companies that are developing these platforms, and we can apply them, and they'll be willing to apply them to TBRS as they get better at those technologies, and we show them that it's really worthwhile. And so that's the roadmap as it is. We're not there yet, and it's not going to be something that's going to be tried in the next year or two, but we're moving very fast, and so it's hard to tell you just how long it would be before a gene therapy like this might be happening. And of course, there's some sort of go no-go points in terms of whether or not it's effective that might tell us that we really should be pursuing this, or we should be thinking about other treatments. So I know I talked a lot, but it's probably longer than folks wanted, but I'm happy to stop there and to take any questions. I hope I covered sort of people's broad interests, and but again, I really want people to not walk away with a question in their mind, so please speak up.
Well, Dr. Gable, that was incredible. Unfortunately, your face is gone, but instead of wasting time with having you leave and come back to the meeting, I think, oh, there you are. Great.
I just had to turn off it on my camera.
Yeah. So first, I just want to applaud you because, the primary audience for this meeting was our families, and you did a really exceptional job of explaining very complicated science in such a wonderful way. So I think cheers to you. Thank you for really doing that so well today.